Welcome to the inaugural Summer Science Café, and of course the inaugural Physical Distancing Café as well. So I'm hoping you'll think of us as takeout food for your mind. Before I forget, let me go through a couple of administrative things. This webinar is being recorded and it will be posted on various social media platforms on the Faculty of Science website after the event is done. So if you knew how to register for this, you should be able to find the link to the recording. As well, for those who are on my Science Cafe mailing list, I'll send the link on to you. I'm Pam Wolf. I teach at Carleton in the Faculty of Science. I teach chemistry and environmental science. I've been at Carleton since 1985. I've been teaching there since 1990. And I've been hosting Science Cafe for about 5 years now. Science Cafes were founded in 2008, so we're going into our 12th year, by the then Dean of Science, who wanted to give Carleton faculty members in the Faculty of Science a chance to share their work, their research, and their interests with the local community that supports them. And just a little note, that dean went on to become the president of a Canadian university. So we train our deans very well at Carleton Faculty of Science. In these times, we need more than ever people who understand the need for science, for fact-based information, and even beyond that, the need for both fundamental and applied research. So we're really grateful for all of you who are joining us and, in a very real sense, giving us your support by coming to the Science Cafes. I don't see all of you listed in your icon forms, but I do see some of you. How many people are new to Science Cafe? Raise your hands for me, and there is a purpose to this. If you scroll down towards the bottom of your Zoom screen, you should see a little button that allows you to give feedback. And yes, I'm seeing a whole pile of hands going up. So yes, if you mouse down or hover near the bottom of your Zoom screen, the feedback icon allows you to raise your hands. So lots of new people. How about people who have been to Science Cafe before? Hopefully that's everyone at some point has had a chance to try to find their raise your hand button. I'll come back to that in a few moments. That's great. It's nice to have new people to Science Cafe, and it's certainly nice to have regulars from Science Cafe able to participate in our virtual version. We'll also be recording. I mentioned that before. So if you can't make it or can't, obviously you can make it. If you can't stay for the entire presentation, you will be able to catch the end of it. The talk today is timely, of course, and that's not a coincidence. We've selected Science Cafes that should be of particular interest to people in these times. But I hope it's also going to make clear the link between fundamental and applied research. There are researchers all over the world right now madly studying SARS-CoV-2, the virus, and COVID-19, the illness that it causes. And all of them would be the first to tell you that the research they're doing now is based on fundamental research that's been going on for many, many years in a huge number of fields. Virology, of course, but sociology, mathematics and statistics, policy, immunology, law. It's probably a mistake to even start itemizing some of those because I'm sure I've left out more fields that were involved than I've included. I should have said including, but not limited to. So all of that research that goes on before we reach a crisis is what allows us to have the science in place to respond to important crises. Our speaker today has been involved in research that's now relevant to COVID-19 for, in her words yesterday, almost 20 years. I'll point out to you, she started as an infant. Professor Edana Cassol is a member of the faculty of the Department of Health Sciences at Carleton. And for the last 15 years, she's been researching how the immune system responds to viral infections. First as part of her master's degree, which she completed in Durban, South Africa, then her PhD in Milan, and a postdoctoral fellowship at Boston. And then finally, the culmination of her career, her arrival at Carleton in 2014, where she leads the Systems Immunology Lab. So she and her team investigate how metabolism contributes to immune response. And her current projects include improving treatment outcomes for HIV patients, improving the efficacy of viruses that are used in cancer treatment, and investigating the mechanisms that drive the inflammatory response of COVID-19. So today she's going to talk to us about how the immune system does interact with viruses. So Edana, we will be switching over. And there we are.
Great, thank you, Pam. So we just have a little bit of a delay while I have while I'm able to share my screen. Okay. There we go.
Oh, and naturally, I forgot a paragraph of my introduction. So while Edana gets her screen in order, I did want to mention that we'll be taking questions at the end, and I'll explain in a bit more detail then. But if you have questions during the presentation, please feel free to type them into the Q&A at that time. Since we're all new at this technology thing and switching back and forth, we are going to wait and address them at the end of the presentation. But for goodness sake, be putting them in now. And other presenters will, other participants will be able to see the questions, and they can also indicate if they have the same questions. So don't wait till the end to type it in. Feel free to type it now if you think of it. And now, Edana.
Okay, so can you confirm that you see my slides? Okay, great.
I do.
Thank you. So yeah, welcome. Thank you all for spending your relatively warm Tuesday afternoon watching this presentation. I've given some form of this presentation many times before, but this is the first time I've actually had the opportunity to do it in the context of a pandemic. So this shall be interesting, and I imagine that a lot of the questions may be focused on COVID-19. Obviously, at the end, I'm happy to try to answer as many questions as possible. Please be aware that this is still a very active area of investigation, and I will try my best to answer based on my current knowledge, okay? I just want to, there we go. Okay. So COVID-19 has changed everything, really. It's changed the way we live. It's changed the way we work. And it's really brought infectious disease to the forefront for a lot of individuals. Up until COVID-19, a lot of individuals didn't really consider infectious disease a significant issue, especially individuals in high-income countries, Canada, the United States, Europe, Australia. And this is in large part because we don't tend to experience infectious disease. And this is just a sort of an infographic put together by the Mayo Clinic. They regularly assess on a yearly basis Americans' attitudes and behaviors and sort of thoughts towards health. And this is some of the data from the 2016 survey. And it really was to see what Americans felt were the greatest challenges with regards to maintaining their health. And you can see that none of the greatest challenges included infectious disease. So all of those greatest challenges were really those associated with non-communicable disease. So more lifestyle-based or genetic-based diseases. So things like cancer, obesity, neurological disease, diabetes, and heart disease. And what's interesting is that at this time, so in 2016, we actually that's when we were experiencing one of the Zika outbreaks, and very few individuals were concerned about the potential of Zika. And this, again, like I said before, is really because deaths from infectious disease have dramatically declined. Again, as privileged individuals in high-income countries, we tend to not experience significant infectious disease. So we may have the flu, we may have a cold, we may experience other types of infectious disease, but as a generalization, we don't suffer the worst of infectious diseases. But even on a global sort of basis, overall, there's been a dramatic decline in the overall rates of infectious disease. And this has really been because of a variety of advancements in medicine. But there are three key advancements that sort of play an important role, or are thought to play an important role in this decreased sort of severity of infectious disease. The first is improvement in sanitation and hygiene. And when we're talking about hygiene, we're talking about everything from hand washing to coughing into your arm, from sterilizing all the way up to sterilizing surgical instruments after and before surgeries. And when we're talking about things like sanitation, we're talking about sort of infrastructure like the sewer systems, which allow us to, in most major cities, remove and purify and clean waste from the environment. The second other major discovery is obviously the discovery of antibiotics, which some of you may be familiar with. The story, the serendipitous discovery of antibiotics. But obviously antibiotics are used to treat, and they're, up to this point, they've been very highly effective at treating bacterial infections and have dramatically decreased rates of severe bacterial infections. The third has really been not only the discovery of a variety of vaccines. So right now there are about 14 different diseases which we do not consider significant public health concerns because we have vaccines. And more than just the discovery of the vaccines has been the universal rollout of childhood vaccination programs, which prevent a lot of these diseases. But the question really becomes, and a question that perhaps is even more pertinent now, is should we be more concerned with infectious disease? So there's relatively low rates of infectious disease. Obviously, okay, this is sort of in the absence of COVID. COVID is a new beast of its own being. We're learning from it. Hopefully this is going to inform and change public health policy moving forward. But in a more general sense, should we be more concerned with infectious disease? And I guess I highlighted concerned just because maybe concerned isn't the correct term. Maybe should be more aware about infectious disease, because even though in high-income countries we have a relatively low risk of experiencing infectious disease or contracting infectious disease, it is still possible. And it is important that we know this to prevent any possible infection. So the graph here really just shows so it's looking at the rates of STIs, so sexually transmitted infections in Canada, basically from 2007 to 2016. And you can see that even though we experience relatively low rates of STIs in Canada, that the rates overall have been increasing in various age groups because people are not protecting themselves as they should. There's different behavioral changes that are happening in our population that are increasing rates of these diseases. So things like syphilis, chlamydia are increasing, especially in some of our young populations. And again, this is a graph that was published a while ago now, so it was in 2004, but it really does show you that at any time, we do have a variety of infectious disease outbreaks happening around the world. And I did include a link here. It's called healthmap.org. And if you actually want to go to this website, you can link in real time to today, what's happening, what outbreaks are around, where do they exist in the various different countries, how many people are affected. So it's actually a really interesting source of information if you want to have a sort of real-time understanding of what various outbreaks. If you go now, obviously there's a variety of, the vast majority of the world is covered in hotspots related to SARS-CoV-2 or COVID-19. But there's still a variety of different hemorrhagic fevers and other types of infections that you can see there where there's small outbreaks around the world. And again, this is a little bit of an old graphic, but it does really sort of emphasize that there's still a number of infectious diseases that we are experiencing as a global population. And these are not only new infections, so infections that we hadn't previously seen in the human population. They're also older infections that we are re-emerging infectious diseases, which we thought we had for the most part previously eliminated, either through and mostly through the introduction of medications or vaccines, but that there are starting to be increasing rates of some of these re-emerging infections as well. And if we look at some of the, sorry, I'm just going to minimize the window here. If we look at some of the most significant recent outbreaks, so this is really a graphic showing from 2002 to 2015, you can see that there are a range of different outbreaks that have been occurring over the last 15 to 20 years that range from old pathogens to new pathogens. So we're talking about, again, old pathogens or re-emerging infectious diseases. We're talking about infectious diseases that have been found in human populations for hundreds of years. So things like cholera or measles or tuberculosis. So we do continue to see outbreaks of these re-emerging infections. So in the context of cholera, it's usually a loss or a breakdown of infrastructure supporting sanitation. So for example, the cholera outbreak in Haiti in 2010 here was really associated with that massive earthquake that happened in Haiti, which resulted in a complete disruption of the sanitation system and then sort of widespread of cholera. Measles is another re-emerging infectious disease. We have a vaccine. Most of us have been vaccinated against measles, but in countries such as the Democratic Republic of Congo, where there is political instability and a loss of healthcare infrastructure, these vaccine programs can't always function as they should. And there are, because of this loss of structure, the loss of vaccinations, we're seeing increasing rates of things like measles. So for example, this is an ongoing outbreak in the Democratic Republic of Congo, and in 2020 alone, there have been over 6,000 deaths associated with measles in this country. The rest of the pathogens, the rest of the outbreaks, are mostly associated with newly identified viruses. So we have everything from SARS, we have things like MERS, the Middle Eastern respiratory syndrome, as well as Ebola, chikungunya, Zika. All of these are newly emerging viruses. And it really is an important question to ask is, why are we seeing these increasing rates of infectious disease? And it's a complicated question. There's not one answer. There's probably a variety of different factors contributing to these increasing rates. It might vary. So the reason why you're seeing increasing rates of different types of disease might depend on the disease, where it comes from. But if we want to generalize a little bit, these are sort of the 5 main factors that are driving a lot of this reemergence. So climate change and environmental changes. So yes, we've heard a lot about climate change in the recent past, thinking about spread of infectious disease with increasing temperatures. We're talking about an increased range of some of the vectors that carry infectious disease. So things like mosquitoes and ticks. In particular, in the Ontario region, over the past 5 years, we've seen a dramatic increase in the rates of Lyme disease. And this really has to do with the survival of ticks and sort of the spreading of their sort of home environment. Antimicrobial resistance is an issue across a variety of different types of drugs, but particularly in the context of antibiotics. Misuse, overuse of antibiotics has led to the development of bacterial strains that are resistant to the treatment, and therefore increasing rates of disease associated with these infections. Human behaviors. Obviously, this is a very complex component. It's not only about sort of high-income countries not really being aware of infectious disease and behaving and not sort of adopting the right behaviors to prevent infectious disease, but it's also things like vaccine hesitancy or not being vaccinated that is contributing to these increasing rates. The other thing at the level of individuals is susceptibility to infection. So as a planet, we have an aging population. As we age, we become more susceptible to infection. A lot of these non-communicable diseases like cardiovascular disease and diabetes actually increase our susceptibility to infection. So this is also contributing. But what seems to have the most immediate and dramatic effect seems to be globalization, economic development, as well as land use. And especially in the context of economic development and land use, a lot of the farming, especially into sort of the more remote regions of the world, where you're increasing contact between humans and animals, and particularly things like bats, you're starting to see these zoonotic events happening. So what is zoonotic? And a zoonotic event, it's the transmission of an infectious disease or an infectious pathogen from an animal to a human. And this is really where we see a lot of these new but also established infections originating in. So we see things like Ebola, Marburg. We think that the SARS-CoV-2 originated in bats, but we're not 100% sure yet. We think about some of the traditional foods. So we've heard terms like avian influenza or the swine flu. MERS is actually transmitted from camels. So the increasing land use, the economic development, the increasing interactions between humans and animals is also starting to increase the rates of infectious disease. So I study viruses, so I may be a little bit biased, but I think, based on some of the epidemiology viruses are of a particular concern with regards to new and emerging viruses. And viruses are interesting because they're acellular, so they're not a cell per se. Instead, they are really just proteins surrounding nucleic acids, which encode for their replication. So a virus cannot exist for an extended period of time outside of a host cell. And I'm sure we'll have some questions about that at the end. But what happens is that to survive, to replicate, the virus actually has to enter into the host cell. And then in the cell, it actually hijacks the machinery of that cell to promote not only its survival, but its replication. And what's really unique about viruses is that they replicate at relatively high levels in some cells. And this replication process, so as the virus is reproducing itself, it's actually quite an error-prone process. So it allows for the introduction of genetic mutations within the virus, which could provide some ability for that virus to improve or escape immune responses. So it's important to note here that most mutations, when you're talking about genetic evolution, actually aren't of benefit to the organism. But in this context, if you do have a mutation that conveys some sort of survival, it will allow for that improved virus to replicate at higher levels to survive. And in the context of especially viruses, they're often evolving to escape the host's immune response. So the question becomes, there's a number of different strategies that we can use to prevent being exposed to viruses, but what does our body do to fight viruses if we are exposed? And this is a big part of what I'm interested in and what my lab studies. And we'll talk a little bit about that in the end. But I really wanted to focus, at least for this talk, given this forum, a little bit more about the generalities. So before we can talk about the specifics of how viruses are recognized, I think it's important to acknowledge the complexity of the immune system. Yes, immune cells, and it's generally thought that the function of immune cells is to recognize between or differentiate between self and non-self. So in order to eliminate and destroy an infecting pathogen, so again, a pathogen is just a microorganism or some other invader that causes disease within the host. Obviously, the immune system has to be able to differentiate between self and non-self. So it has to recognize a pathogen as foreign in order to destroy it. But it also, at the same time, these cells have to recognize that host cells are not foreign and that they should not be destroyed. But when we delve into the specifics of the immune system, it's actually so much more detailed than that. And in my opinion, so much more cool than that. You think about the ability of the immune system to, and the functions and sort of the specificity. So you think about that every time you're infected with a different type of pathogen, a different type of immune response is induced. These responses are very specific to the pathogen to ensure, or to hopefully ensure, the optimal response to remove that pathogen. So depending on if you have maybe a more bacterial infection, you want to induce a really strong immune response to target the bacteria which generally exist outside of cells. Whereas, for example, in a viral infection, viruses live within cells. So you have to have a different type of immune response that targets and destroys infected cells. So, you know, some of those subtleties and the reasons and the ability of the immune system to do that is actually very interesting and cool. There's also it's more than just being able to recognize self and non-self. It seems like, and it's been shown multiple times now, that the immune system knows when to respond to foreign invaders and when not to respond to foreign invaders. So for example, I'm sure most of you or many of you have heard the term microbiome. The microbiome is all of the microorganisms that exist in and on our body. Most of them are good bacteria. Sometimes it's viruses, sometimes it's also fungi, but the vast majority is bacterial species that actually interact with us and provide some level of health. And only when you have the establishment of bad bacteria, pathogenic or disease-causing bacteria, do you really start to have disease. But on a day-to-day basis, we're exposed to millions of different microorganisms, and our immune system does not respond. So the ability of the immune system to know when to respond and when not to respond is actually really cool. And if you also think about some of the other foreign molecules that we ingest, whether we're talking about drugs, whether we're talking about antigens from the environment, so proteins that we take in from the environment. You know, the vast majority of us don't sort of induce strong immune responses in these contexts. So the immune system has these very specific mechanisms to regulate this. And it's even more complex than that, because immune cells are found in every tissue of the body. And in each tissue, they have a very different function. So in the absence of an infection, immune cells play an important role in just maintaining the integrity of a tissue and the function of a tissue. And then if an individual is exposed to a pathogen, or if they are infected by a virus or bacteria, the immune cells are then able to initiate the immune response. So you can see here, this is sort of the gastrointestinal tract, and you can see it's a cartoon of the structure of the small intestine. So this is what it looks like in your intestine. So this is the lumen of the gut. So this is what's the external environment that, you know, when you're consuming food, you're metabolizing food, you have all of these bacteria in this environment, these commensal bacteria that play an important role in these processes. And right below the surface of the epithelium in the gut, you have all of these immune cells, which, even though there's all these bacteria in the environment, are generally not responding. They can be activated really, really quickly if they need to be, if there's a bad bacteria that's introduced. So even the organization of the immune cells within a tissue is complex, but plays a critical role in their ability to mount a functional immune response. So I don't want to get too much into the details. Again, thinking about, maybe you guys are all coming from very diverse backgrounds, but in order to understand how the immune system responds to and how we sort of defend against viruses, it's important to have a little bit of an understanding of the immune system. Again, this is not an immunology class, but I just wanted to give you some of the basics. So when you're thinking about the immune system, there's really sort of two lines of defense. There's the first line of defense, which are really those soldiers at the front of the exposure that respond quickly, that try to clear the infection before the infection can spread throughout the body. And these responses are induced in the matter of hours. And this group of cells are called the innate immune system, okay? The second line of defense takes longer to be induced because it's more specific, and there's a variety of different signals that have to be induced, but these are the very potent killers and destroyers of microorganisms and infections. And this second line of defense is called the adaptive immune system, or adaptive immune response. And so what happens in the context of a viral infection, you can think about your particular virus of interest. So let's talk about SARS-CoV-2. So you would have the SARS-CoV-2 virus. Again, SARS-CoV-2 is the causative agent of COVID-19, enters into the lung, and it starts to replicate in the epithelial cells. So the layer of cells on the surface, sort of on the outside of the lung. And what happens is it replicates in those cells, and it causes some of those cells to die. And those cells release signals into the environment, which start to activate the innate immune response. And some of the first cells that are activated in that environment are called macrophages. And these cells, which are the cell, I will acknowledge these are the cells that I study, so I'm probably most interested in these cells. They actually initiate the immune response. So not only do they respond to the signals that have been released by the infected cells, but if they also come into contact with the virus, it can actually recognize the virus and turn on a sort of functional cascade that induces subsequent immune responses. So what do? They start producing all kinds of proteins that turn on the antiviral programming, so the immune response against viruses in other cells in that local environment. And they also start producing a lot of inflammatory molecules and mediators that recruit other types of immune cells to that initial site of infection. So again, if we're talking about SARS-CoV-2, this would be an increased recruitment of these cells to the lung. And again, there's a variety of different cell types that are part of the innate immune system. Some play a role in viral infections, some don't. But one of the key cells that play an important role, at least in the initial control of viral infection and destruction of virally infected cells, are the natural killer cells. And I mean, based on the name, they have one of the coolest names of all of the cells of the immune system. You can sort of surmise or deduce their function in that they kill infected cells. So after the macrophages get activated, the NK cells get recruited to that site of infection, and now they're able to, if they come into contact with an infected cell, destroy and eliminate that cell. And the reason we want to destroy and eliminate infected cells is obviously to prevent the further replication and propagation of the virus. So while all of this is happening, we also want to induce the adaptive immune response, because while the innate immune response tries its very best and is usually quite effective at destroying and eliminating viruses, it's usually not enough to completely eliminate the infection. So at this time, what's happening while all of those innate pathways are activated, we also have the steps happening to induce and activate the adaptive immune response. So again, this is driven mostly by two types of innate immune cells. So we have the macrophages as well as the dendritic cells, which are always just taking in sort of things from the environment. So for example, the dendritic cell is going to be sampling anything it comes into contact. And what it's going to do is digest the virus or whatever comes into contact and display it on the surface of the cell. And by displaying it on the surface of the cell, it actually acts as a flag to say, okay, adaptive immunity, you need to be turned on to induce a functional response to target this pathogen. So the dendritic cell takes up the virus, it homes to the lymph node. So if anyone's had an infection where you've had your lymph nodes swelling up, part of that is because of the induction of the immune response. But anyway, you have the dendritic cell taking the flag saying, we have an infection, there's a virus, this is the virus that you need to mount an immune response. And those are the signals that are required to turn on the adaptive immune system. And then again, it takes several days for this process to happen. But then when it works incredibly well. And there are two cell types that really drive a lot of this defense. There's the B cells. I don't know if any of you guys have heard of B cells before, but they play an important role because they produce antibodies, which is a protein that is produced and targets and binds to the pathogen or the virus to allow it to be cleared and eliminated from the body. The other thing that's really critically important in, especially in viral infections, because the virus exists within cells, is to activate the cytotoxic lymphocyte response. So cytotoxic, cyto is cell, toxic meaning toxic, to destroy. So these are cells that kill other infected cells. So they have a function that's similar to natural killer cells, but they're more specific and more targeted. And it's actually these cytotoxic lymphocytes that kill and destroy not only virally infected cells, but also cancer cells in the body. And so I have a cool little video I wanted to show you guys. It's about 2 minutes, but it shows you how these CD8 T cells are killing. And again, this is in vitro, so it's in a culture system, but it shows you how effective these cells are at destroying it. It uses cancer as an example, but again, the same sort of thing happens when it's targeting a cell that's infected with a virus.
Inside all of us lurks a serial killer. A killer whose primary function is to kill and then kill again. These are cytotoxic T cells, a specialized member of our white blood cells. They patrol our bodies, identifying and destroying virally infected and cancer cells, and they do so with remarkable precision and efficiency. There are about 5 million T cells in a teaspoon of our blood, engaged in the ferocious and unrelenting battle to keep us healthy. These amorphous blobs move around quite rapidly, pushing out their leading edge and probing their environment as they go. When a cytotoxic T cell finds a cancer cell, membrane protrusions rapidly explore the surface of the cell, checking for the telltale signs of cancer. They kill their targets using poisonous proteins visible here in red. These cytotoxic granules move down special pathways in the cell called microtubules to the interface between the T cell and the cancer cell. The T cell punctures the surface of the cancer cell and delivers its deadly cytotoxins. This is very important in our bodies, where cells are packed together, as it focuses the lethal hit on the target and minimizes collateral damage to the neighboring healthy cells. The fate of the cancer cell is sealed. The T cell then moves on, hungry to find another victim.
Okay. There we go. So I recognize I want to leave lots of time for questions. So I just want to end it with a little bit of discussion around what determines the outcome of a viral infection. We know depending on what type of virus that you're exposed to, some people may survive, some people may succumb to the infection. So it's really important that we understand some of these processes. So you think about once a virus enters into the body, the battle begins. Especially in that early stages of infection or acute infection, when it's the innate immune response targeting the virus. It really is a back and forth bi-directional communication between the two. The immune response tries to destroy the virus mutates potentially to escape that response. And then depending on sort of which aspect wins, if the immune system is more successful, then the individual can recover, clears all the virus from the body, and then survives, obviously. So again, the outcome of a viral infection very much depends on our ability to respond. So when a virus enters the body, the battle begins. The immune system tries to fight it off. If the immune system is successful, then the individual recovers from the infection. However, if the virus is able to evolve, mutate, and escape, it's possible that either the virus wins and the individual may succumb to the infection, so they may die, or it may enter into this sort of state of chronic infection, which is sort of this long-term infection where the immune system and the virus is replicating at relatively low levels because the immune system is able to regulate it to some degree, but that it's a continual low-level battle that exists. So these are things like when you're talking about HIV infection, you think about how someone can live for up to 10 to 15 years without even sort of having the consequences, it's the severe consequences of disease. Hepatitis is another example. But it's important to recognize that especially in viral infections, the immune response has to be tightly regulated. And this is really because whatever the immune system is doing to try to not only destroy infected cells, but to clear the virus, there's also potentially negative consequences of these molecules, these proteins on the host and the host's tissue. And that's why when we're talking about viral infections, it's important that we need to have a tightly regulated immune response. And there's good and bad. So for example, in the context of Ebola, I don't know how much of you, it was in the news the past couple of years, and there was a recent outbreak even this year. But you think about the sort of the disease and the stages of infection. So usually someone who's been infected doesn't show symptoms for the first 10 days. And then when they start to show symptoms, it's because there's enough virus in the body that the immune response is strong enough to start to respond. So things like in the first 10 days, they get fever, fatigue, headache. And then as the virus continues to replicate in the body, the disease becomes more severe. So the consequences become more severe. So you end up with things like diarrhea, chest pain, coughing, eye inflammation. And then by about 2 weeks, so 15 to 17 days after infection, you have some of the more, about 18% of the patients start to have this more hemorrhagic type fever where you have bleeding. And what's interesting is after various rounds, like various researchers being involved in this, it's actually not the virus per se that's driving this damage and this sort of this vascular response. It's actually the immune response. So what happens is that the macrophages actually ingest the Ebola virus, and that triggers the macrophage to be activated. So what they do is they trigger all of these clotting cascades, so that increases coagulation of blood. And it actually reduces blood supply to the organs, and that's what can often contribute to some of the hemorrhagic features of infection. It also produces a lot of the macrophages, once they ingest the virus and become activated, produce inflammatory molecules, which can damage the blood vessel lining and things like that and cause them to leak. So it's not actually the virus, it's the immune response that's causing the disease. And what they think is that COVID might actually have a similar type of process. Again, I talked about this a little bit. So you have the virus entering into the lung, infecting some of these epithelial cells that line the outside of the lung. And then as they become infected, they are destroyed and they release these signaling molecules into the environment, which activate the macrophages. And the macrophages then recruit all of these other immune cells into the lung. And these activated cells release more inflammatory mediators. They alter sort of the different types of cascades, which causes damage and destruction to the lung. So there's a number of different immunotherapies that are being used to try to limit inflammation in the context of COVID-19. But the problem with targeting sort of a broad immune molecule is that you also increase then that individual's susceptibility to other types of infections. But again, if we can understand how the macrophages and how other cells play a role in this chronic inflammation, maybe we can find a way to not only limit inflammation, but prevent the fibrosis and the damage to the lung. So in the absence of a vaccine, so again, this is particularly important in the context of COVID-19, but a lot of other viral infections, which we don't have vaccines for. What can we do? What treatments are available? Again, in most of these viral infections, we don't have a treatment necessarily to target the virus. So in some cases we do, but for example, in COVID-19, most of the time we're just trying to deal with the symptoms rather than to clear the infection. So my research, again, is very much related to this. So we really want to look at the interactions between viruses and the innate immune response, and specifically macrophages, to understand and to develop smarter responses, to find mechanisms and way to fine-tune immune responses so that we only have the components of the antiviral immune response that we need, and we can shut off in a timely manner any of the components of inflammation that might be destructive and damaging. So what we want to answer are things like, what makes a successful immune response and how might that differ if a cell is exposed to different types of viruses? How does the virus reprogram the macrophage or the function to escape immune recognition? And then what eventually contributes to the immune response becoming disease-causing or pathogenic? And again, I don't want to take too long, but a lot of my research focuses on the potential role of metabolism. So when you think about metabolism in a cell, you think about all of those complex pathways that are important in driving the energy and the function of the cell. So yes, your cells need energy to function. You also need to be able to produce things like proteins and nucleic acids and fats to maintain the structure and the function of the cell. But what's increasingly recognized in the context of the immune response is that very specific pathways are activated in the context of different types of infections. And the question really becomes, if we can figure out if those pathways are in fact driving the function of the immune response, is there a way to target metabolism to prevent certain aspects of immune responses from having, or to improve or increase antiviral immune responses? And again, I'm happy to take questions at the end. My focus is a lot on the mitochondria. I don't know how much you guys know about the mitochondria, but it's really a central organelle in the cell. It's important in energy metabolism. It produces the vast majority of energy in the cell. But what's increasingly recognized is this organelle within the cell also plays a really critical role in antiviral immune responses. So not only in producing the energy required, but producing all of these other molecules that can activate immune signaling and drive immune function. And this is, so we have sort of a three-pronged approach. We can actually isolate cells and culture them in the lab, so in vitro, and we can infect those cells with various types of viruses, and then we can understand how the immune function is related to the metabolic function. We can also use ex vivo models, so we can infect animal models with various types of viruses, and then we can isolate out the different tissues from those animals, and then we can actually look at how the immune responses may differ in different organs. And then the idea is once we understand what a normal or a healthy immune response should look like, we can actually get clinical samples from patients. So this might be blood from HIV infected individuals, or it might be tissue samples, like one of our projects is looking at the role of bacteria driving chronic wounds. So there's various different types of tissues or disease samples that we can get to see and compare the responses in the disease compared to what we would expect as a healthy response. And that helps us to identify which pathways may be altered and potentially could be targeted for therapeutics. So this is just the last slide, really. This is just an example of one of the projects where we're looking at the effects of HIV infection in macrophages in the brain. So what we can do is actually obtain human brain macrophages. We can infect them with HIV, and then we can look at the differences in the function in infected cells and uninfected cells. And then what we can do is use these findings to better understand what happens in patients, because then we can get brain tissue from individuals who have infection with and without neurological disease, and look at if these pathways are altered in these patients as well. And these are the graduate students in my lab. They're all working on different projects. Some are working on COVID, some are working on HIV, some are working on wound healing. So again, if there's interest, I can go into it more. Okay, and so I'm open for questions now. And I'm really very, very sorry about the interruption.
Yes, we'll get our kinks worked out so that when you all come back for our future science cafes, which I'm confident you will, hopefully our technology will be smooth. We'll be opening to questions in two forms. I'll start with some of the Q&A typed questions that I've received already while Edana organizes herself, but we have someone in the background as well who will be monitoring for people putting their hands up. So it will be possible, if you have a complex question, to ask it verbally. First, you'll be given, that sounds so terrible when I put it this way, but you'll be given permission to speak by one of the hosts in the background. Then you will have to unmute yourself, but you'll receive a message to unmute yourself. What I would ask you is that you limit this to specific questions though, rather than making statements, etc., because we're low on time. So we'll try to keep the questions focused. I'll start with two that I'm going to group together that were in the Q&A, which is, can we boost our immune system, and to what extent can nutrition influence the efficacy of our immune response? Because I'm going to assume those are linked questions, at least to some extent. So nutrition, but anything else as well that could boost the immune system.
Right. So there are a variety of ways we can boost our immune system. Nutrition is an interesting idea. Obviously, as someone who studies metabolism, what nutrients are available in the environment very much drives and regulates the magnitude of the immune response. I think having all cells require energy. Having well-balanced diets is probably the best approach. There's no specific magic nutrient or molecule at this point that we're aware of that we know is like the end-all be-all of protective molecules. But I think having obviously enough nutrients and well-balanced nutrients will help you fight infection. It ensures that your immune cells have everything that they need in order to respond. With more specific, I don't think we know enough at this point to identify specific nutrients which would best enhance responses.
Okay, and another one that I have written here is, if COVID entry is generally via the mouth, nose, and eyes, why doesn't public health tell us to wash our faces as they tell us to wash our hands? And then linked to that, if soap breaks up the membrane, as the questioner has read, as have I, read that is, I don't know these things, what is the difference between using hot and cold water? Some public health people are saying when you wash, you should use cold water. Others are saying not.
So I think, so soap is the most effective way because it actually breaks up the membrane of the virus, the lipid membrane, so it disrupts that. When you're using soap, I think, again, I'm not a public health expert per se. I think that it should be enough whether you're using cold or warm water to do that, as long as you're using sufficient soap and you're cleaning the hands to the best of your ability, you're completely cleaning your hands. The idea with hot is that if you add that it introduce an additional stress, it could potentially affect the stability of the virion, the virus, and help in that way. I don't know that they've done it conclusive enough studies at this point to prove that hot water will do that, but that is the general idea. If there's any way that you can improve or disrupt the virus integrity, that's a way to do it. So the question about washing the face, I guess the issue is more that infection occurs through mucosal membranes. So if you're exposed on your skin, you won't become infected. But if you get exposed on the, in your nose or in your mouth, that's where the higher potential is to get infected. So the idea behind washing your hands is because people tend to put their hands, you end up with virus on your hands, and then you're touching your nose, you're touching your mouth, you have an increased risk of transmitting it to those highly susceptible mucosal membranes. Your skin in general will not allow the virus to enter into the body.
Okay, and I do have one more written down here, which was where are we in understanding whether we get community or herd immunity post-COVID?
So that's a very active area of investigation, and that's been prioritized by our government as well as many other countries' governments. I think there's promising results to show that there may be more. Originally, there was no proof to show that there was long-term immunity induced. I think there's a number of studies now that show that there are these memory immune responses that are induced, which again, the adaptive immune response is able to sort of have these long-term memory responses, and that's why vaccines work. And that's why if we're re-exposed to a virus, we can protect against that virus. So it's promising. I think that the mechanisms and the length of the memory is still a sort of an active area of investigation.
Okay, the next one I'm seeing here is it possible to suffer a deficiency in the adaptive immune system cells, making those people more susceptible to viral infections, cancer, etc.?
Absolutely, and there's a variety of different, so there's genetic defects that so if you have a genetic defect in any of the molecules that are part of those adaptive immune pathways, you will have altered an adaptive immune response and be more susceptible to infection. If you're nutrient deprived, you can potentially have alterations in immune function. If you have other types of infections like HIV, they can make you immunosuppressed. Certain drugs like corticosteroids and stuff make you immunosuppressed. So absolutely, yeah.
Okay, how are you studying immune response? What type of experiments? Very interesting. Comment.
Very interesting. So again, we use sort of two different approaches. We're actually able to isolate the cells from humans or from mice, and we're actually able to infect those cells in the lab, and we're able to study how that infection reprograms the metabolism of the cell. The other thing that we can do is we can actually get tissue samples from patients. And then what we can do is we can look at, in those patients with whatever viral infection we might be interested in studying, how the metabolism is altered as a consequence of disease. So we use things like, again, without getting too technical, because I think it's beyond the scope of this, we can look at the single cell, we can look at how the immune function is changed, the types of proteins that are produced, the metabolic pathways that are turned on and turned off. So again, if it's something you're really interested in, send me an email. If you want to go to the Health Sciences website, you can get my email, and I'm happy to answer more details in that way.
Okay, got one. This may not be something you could answer, but out of curiosity, there are a lot of immune boosters, supplements on the market, especially now with COVID, they're highly marketed. Do immune boosters do anything to help the body not get sick or to help the body fight off cold or flu? Or is it just a marketing scheme to sell products? Is there any truth behind them?
So there is probably some science behind the sort of provision of certain types of nutrients and boosters. The problem is that the way that you're taking most of those boosters, the majority of it is being metabolized and excreted from the body before it can have any beneficial effect. And depending on what form they come in, you may not be receiving the amount that's required to actually have that beneficial effect. So yes, they're mostly just hype. I would say it's more important to make sure that you're properly nourished, that you sleep well, that you're not particularly stressed out as much as possible, because stress suppresses the immune system. It's more about maintaining your overall well-being to ensure that your immune system has the best possibility to function at its optimal.
Okay, can you comment on the impact of diabetes on responding to COVID-19, and as well whether and how vitamin D aids the response?
So there's a lot of emerging data to suggest that vitamin D might be protective. Diabetes, any type of chronic disease, tends to have low levels of inflammation, which can, in the long term, disrupt and affect immune function. In diabetes, also the addition of high levels of glucose in the blood can also alter and affect immune responsiveness. So the combined sort of aspects of that chronic disease, the chronic inflammation, and the altered immune responses seems to have and drive a lot of the immune deficiency associated with diabetes. Again, with vitamin D, I am not 100% sure with regards to the mechanism as to how it works to improve immune function. I know for a while it's been proposed that vitamin D is beneficial to the host and can be protective. I don't know the actual mechanisms, I'm sorry.
Okay, how do antiviral drugs work, and to what extent are they effective in the case of COVID-19?
Sure. So antiviral drugs are drugs that specifically target the virus. For the most part, when you're talking about antiviral drugs as a class, you need to be talking about drugs that specifically target each individual virus, unless there's some mechanism in the replication or in the propagation of the virus that's sort of consistent across a variety of different viruses. So some of the drugs that they're talking about, the antivirals that are being used in the COVID-19, how they work is by preventing the replication of the virus. So they inhibit a very specific component of the transcriptional response, so like the replication of the virus, to prevent the spread and the sort of release of the variants into the body. So the idea with antivirals is you're targeting the virus. The problem with that is that viruses can escape, they can mutate, and they can potentially evolve the ability to no longer be responsive against antivirals. I keep stumbling because I want to say antiretrovirals because I study HIV and that's a specific type of antiviral drug. But the idea is that my approach is to target the immune system because you cannot develop resistance against your immune system will not develop resistance. So it's an alternative approach to try to treat the disease.
Okay, how does metabolism function in our present age of widespread use of anti-metabolites?
I mean, we're using them in a variety of, I think we're just starting to understand. I mean, I can only answer it in the context of metabolism or in the context of the immune response. It very much depends on which anti-metabolite that you're talking about, because each metabolite, not every metabolite has a specific consequence of immune function, but some have more important roles in immune function. So for example, some of the metabolites of the Krebs cycle, so the TCA cycle, are very potent signaling molecules. So it's not just about energy, it's also about the activation of the signaling cascades that are required to induce an immune response. So again, it depends, it's a little bit of a broad question. It depends very much on what anti-metabolite you're talking about, the metabolite that's targeted, and the potential consequences. But things even like metformin, these more general modulators of metabolism, it's increasingly recognized that they have quite pronounced effects on immune function. And we're really just starting to understand what the consequences of that might be.
And this may be linked. There are many mitochondrial dysfunctions. Last I read was 1 in 500. How is this affected by the widespread use of anti-metabolites?
Anti-metabolites. Again, I can talk to it more in the general. So yes, there's a variety of mitochondrial diseases that are generally associated with a mutation in some form or component of mitochondrial function. The idea being that depending on if that component of mitochondrial function is required to generate or mount an immune response, it is possible that it could affect your ability to respond to viruses. So some of the mitochondrial diseases often identified in children, because often the individuals don't survive, especially in the most severe cases. Often they succumb to things like secondary infections. They have an increased susceptibility to bacterial and viral infections, again, depending what component of the mitochondria might be affected.
Okay, how does the widespread use of immune suppressants tie into the recent explosion of infectious diseases?
It's a good question too, because most of the immunosuppressants that we use, whether it's to treat autoimmune disease like rheumatoid arthritis, if you've ever seen those commercials on TV, they talk about increased susceptibility to infection. When you're targeting these pathways, especially because you're usually targeting quite important players in the pathways, you are increasing susceptibility to infection. Whether that's contributing to the overall increase of infectious diseases that we're seeing, not only in high income, but in sort of across the board on the global scale, I don't think it's been directly linked. But as an individual, if you're taking immunosuppressants to regulate things like allergies or autoimmune disease, you are at increased risk of infectious disease.
Okay, what are the challenges in developing a COVID vaccine? And is an overactive immune system as a result of the vaccine a potential danger to the patient?
So you have to remember that vaccines are before you're infected, right? It's a preventative measure in most cases. Most vaccines obviously are tested to ensure that the response is appropriate and functional. I think that there are potential side effects associated with some vaccines, but that is incredibly rare. I think that whatever vaccine may be generated to COVID-19 will be functional and ensure that the vast majority won't induce an over response. Otherwise, it won't be allowed to come to market. I hope that answers the question.
Okay, I'm guessing the immune response depends on the viral load.
Yeah, yes, it would. So obviously the more, I mean, it depends a little bit on which virus we're talking about. We're talking about viruses very generically here. Each virus induces a very specific immune response. So yes, the more virus you have in the body, the stronger the immune response you will induce. And at some point, that's especially when you have high levels of virus in the body, that's when you start to have these overreactive immune responses that can cause secondary damage to tissues and cause disease and things like that.
Okay, how exactly does stress affect the immune response? I think
So
a big question for all of us in these challenging times.
So a lot of the steroid, like cortisol, and a lot of the molecules that are produced as part of a stress response actually directly interact with and suppress the responsiveness of the immune cells. So a lot of it is driven by cortisol directly, but some of the other sort of sterile molecules that are associated with those pathways also interact with and downregulate immune responses.
Okay, since viruses migrate so rapidly, why do vaccines work? I'm particularly concerned about the continued efficacy of any vaccine for COVID-19. Will it cease to be effective as the virus mutates?
Okay, so it's a good question. At this point, again, we have to talk a little bit on each virus. So, for example, we haven't been able up to this point to develop a vaccine for HIV because it mutates and escapes at such a high rate. SARS and SARS-CoV-2, again, the causative agent of COVID-19, doesn't seem to mutate and evolve at the same level. So the idea would be to ensure that whatever vaccine you're using is targeting sort of a conserved region on the virus that would not allow for the evolution of that virus unless it resulted in the virus sort of being destroyed. You can target specific components of the virus that are so fundamental to the functioning of that virus that even if there was a mutation, it's unlikely that that virus would survive. And sort of that's the approach to use. At this point, SARS-CoV-2, it doesn't seem like it mutates as rapidly as other types of viruses. So I think that whatever vaccine is developed, it should work well.
Okay, and immune suppressants seem to be of interest to people. How do you boost the immune system if at the same time you're suppressing it?
So, I mean, it depends on how you're suppressing and it depends on how you're boosting, right? So depending on what target, what drug, so if it's possible you have an overall immunosuppressant, it would have more dramatic effects on immune function, affect a variety of different cells. Again, it depends very much on how the drug works and the target of that drug. But most immunosuppressants target one specific molecule or pathway, and just because you're knocking out that specific pathway, although it's important in the immune response, doesn't mean you're completely wiping out or sort of destroying all of your chances to respond. So again, without knowing what drugs you're specifically talking about, it's possible to ramp up certain components of the immune response while dampening other components. That makes sense.
And this may be a follow-up, I'm not sure. So the widespread use of prednisone, which is synthetic cortisol, is causing immune suppression. Is that leading to a concomitant rise in infectious diseases?
Again, I don't know that there's the epidemiological data to support that on a sort of global scale, but as an individual, for sure. And it's one of the precautions you have to be aware of if you're taking any type of steroid, is that it can suppress your immune system and it increases your risk of infectious disease.
Okay, and the last question I'm seeing still live says, how can we sign up for, oh, another one popped in. We'll do that one last one, and then I'll answer the sign up for the mailing list. And so thoughts on vitamin C and zinc, sorry, my mouse is in the middle, zinc picolinate supplementation for boosting our immune system for COVID-19.
They're sort of along the same, and I know that people are trying to do whatever they can to try to protect and ensure. Again, there is science to support that vitamin C is important in regulating various enzymes and proteins within immune-based cells. Whether taking vitamin C or zinc will have direct effects on protecting you from COVID-19, again, there's not that data to support that. What I would say is that if you're doing everything you can to be as healthy as possible at the level of nutrient, at the level of stress, at the level of sleep, all of those things are going to improve your ability to respond to the virus. But it's so complicated. There's genetics, there's so many other aspects that are involved that, yeah.
Okay, and then the last of the questions was, how do we join the Science Café mailing list, which I had mentioned when I was talking about my introduction. When Science Café meets live, I always put out a sign-up sheet for people. That's not going to work in this format, but if you send an email to the contact person for Science Cafés, which is someone in the office of the Dean of Science who does all of the hard work in the background, I just stand up in front and I'm the public face. But the people who do all the work in the office of the Dean of Science, the contact people when the Science Cafés are advertised, they will pass your email address on to me, and I keep those myself. So I do need to say a disclaimer. They are secure from attacks on the university in a way that an official mailing list would not be. However, they're subject to me being careful, for instance, that because I store them on a database on my own laptop, or a database, excuse me, Excel. They're subject to me remembering to always blind copy and not put people in the address line. I have never forgotten yet, but I could. It's not a mechanized system. It's simply a mailing list where people have entrusted me with their email addresses in exchange for getting information about Science Café, and I send out information about other public events at the university that are of interest to people interested in science. So if under those terms you would like to be on the mailing list, the Office of the Dean of Science will pass your email on to me. But be aware that it is me as a person. I am not, in that sense, a representative of the Office of the Dean of Science. If I were, I would be storing all of these in officially protected university formats. It's people who've given me their emails. So in that sense, a personal relationship sounds a little alarming, but it is in that sense. And if you want me to send you these notices, I'd be happy to, but the best contact is the Office of the Dean of Science, because if I give you my email verbally, which I'm willing to do, pamela.wolf@carleton.ca, and Wolf is spelt with 2 Fs. It's not complex, but it's something that I don't have, a slide for. So Office of the Dean of Science is a good place to start, and they'll pass it on to me. So that wraps up our first virtual summer Science Cafe. I would be willing to call it a success, even though Edana and I were both thrown out of the presentation. So I hope the rest of the participants are more impressed with the two of us and Edana's talk than obviously Zoom was. But we have prevailed, and I hope you'll join us for future Science Cafes. If you ask to be added to my mailing list, I will be sending out the list, and they will certainly be appearing on the website for the Dean of Science, on our social media platforms, etc. I think 2 weeks, and I believe we're migrating to our more usual day of a Wednesday, but certainly you'll be able to get information on that, and I hope you will all join us again. So thank you so much for participating. And Edana did tell you how to contact her at the Department of Health Sciences at Carleton University if we haven't managed to get to your questions. So thank you all very much.